Hello and welcome everyone. Thank you so much for joining us today. My name is Christoph Meinert. I'm the CEO of Gelomics. We are a Brisbane-based biotechnology company focused on advanced 3D cell and organoid culture products. We enable our customers to generate lifelike models of human tissue to advance biomedical research and reduce the reliance on animal experimentation. We're very excited to host our second webinar this year. And today we're focused on 3D bioprinting. We'll discuss the development of advanced extrusion-based bioinks and the biofabrication of engineered tissue and organoid-based architectures. It's my absolute pleasure to welcome our two amazing guest speakers today, Gelomics production manager and PhD student Jordan Davern, and Associate Professor Riccardo Levato, an internationally recognized key opinion leader in the space. We're very honored to have you both join us today. Thank you so much, Jordan and Riccardo. Just a few quick housekeeping items before we get started with today's webinar presentations. If any of our attendees experience any technical issues, please let us know using the chat function in the GoToWebinar software interface. We'll try to address these as soon as possible and fix up any issues that might come up as well. If you have any questions, and I highly encourage you to ask questions to our speakers today, please use the dedicated Q&A feature on the software interface, and we will make sure to address these questions at the end of each of our presentations today. So without any further ado, we'll probably just get started into the first presentation by Jordan Davern. So I'll just give a quick bio. So Jordan is currently completing his industry-led PhD through the Australian Research Council Training Centre for Cell and Tissue Engineering Technologies at the Queensland University of Technology in Brisbane, Australia. His research focuses on developing highly tunable bioinks for the 3D printing of complex tumor microenvironments with precise physicochemical properties to study cancer-associated processes. Jordan completed his Bachelor's of Biomedical Science and his Master's of Science by research at QUT, during which he investigated the formation of bioengineered vascular networks for regenerative medicine applications. Jordan is also our production manager here at Gelomics and has overseen the development and the expansion of Gelomics' current product portfolio with experience in the development of biomaterials, quality control procedures, research and development in general. So thanks again, Jordan, for having us today, or for being here today and presenting today. So we really look forward to your presentation. If you don't mind, I'll just share your screen now, and please accept it, and we get started with your presentation.
Yeah.
And I will turn off my camera just so everyone could focus also on Jordan during this presentation.
Yeah, perfect. Can you hear me okay?
Yes, we can.
Yeah, I guess that. Thank you, Christoph, for the kind introduction. And today I'll be giving you a brief insight into what my PhD titled The Tailorable Gelatin-Based Bioinks with Lapinite for Extrusion-Based Bioprinting. So 3D bioprinting, there is still this main issue with trying to maintain the equilibrium between printability and biocompatibility. And many current inks that are currently used today still fall within this traditional bioink realm. So it's within this medium range of printability and biocompatibility. And there's a current need to then further advance this to create these advanced bioinks, which is the best of both worlds. However, to be able to achieve this, you need to have certain bioink properties, which can include shear thinning, characteristics, viscoelasticity, biocompatibility, and gelation kinetics. A way to actually look at incorporating and achieving these advanced bioinks is the use of nanocomposite bioinks, which is what I'll be talking about today. So in order to be able to have bioinks and translate current hydrogel models that have been used extensively, they need to have certain characteristics, and a lot of the hydrogels that are currently used lack this. They're very Newtonian-like, they have a low viscosity and printability, and you'll most likely end up achieving something like the source bottle, which is not ideal putting into a 3D platform. In order to then be able to achieve these 3D nice constructs and these ideal bioinks, you need to control and maintain these certain characteristics and properties to achieve these nice high fidelity structures. A way to understand bioink properties is using rheology, which is an excellent pre-screening tool to be able to look at certain characteristics during this printing process. It allows you to also look at large rheological added concentrations to find the optimal concentration, and looking at these specific processes that are associated with printing, which can be the shear thinning capacity or being extruded, the shape recovery once it's being extruded, or the yield stress and the sedimentation while within the barrel of the model. The proposed bioink material that we've been looking at is gelatin methacrylate, which has been extensively used within 3D cell culture. It's semi-synthetic material and can be cross-linked via photopolymerization, and has been extensively used not just in 3D cell culture, but also in other 3D additive manufacturing techniques, which can include microfluidics, lithography, and bioprinting. Today we'll be focusing on extrusion-based bioprinting. So I've been looking at incorporating gelatin methacrylate gel with another rheological additive known as laponite. So laponite is commonly used in the cosmetic industry, and if you've ever used a type of skincare such as Nivea, you've most likely used it. It's a nanoparticle and it's a synthetic semite clay, and has previously been used as a bioink additive prior for osteogenic applications. In order to be able to, for our goal for these bioink requirements, we want to be able to maintain the stiffnesses and target the tissues at stiffness specifically. So we want to be able to, for these bioinks, to have a tunable mechanical stiffness range. We want it to be able to be shear thinning. We want to be able to print it at room temperature, which is within 22 and 26 degrees. And most importantly, it needs to be cytocompatible. So laponite has been extensively used previously in prior papers and studies, using for a number of different applications, from osteogenesis to vasculogenesis, looking at large barrel defects. And many people currently use laponite at a concentration of 0 to 8%. So then this led us to our aim: to investigate the use of laponite as a rheological additive to see if it would enhance Gelomics biofabrication, and looking at a specific Gelomics concentration ranging from 5 to 15 to account for the mechanical stiffness range we'd like to achieve, and looking at the lower concentration end of laponite of 0 to 4%. The general methodology focused around building, developing the bioink development, achieving these different concentrations, and then undergoing a rheological profile, looking at 4 main characteristic tests, such as viscosity for shear thinning capability, thixotropy to look at shape fidelity and viscoelasticity, a temperature sweep to look at what printable temperature that we'd like to achieve, and the cross-linking and mechanical characteristics, followed by doing printing characterization of fiber and pore diameter for optimal printability window, and then some CFD simulations in 3D to get an idea of the shear stresses that were occurring, followed by biological compatibility. So begin with looking at the rheology and looking at the viscosity profile, in which we can see here the differences between the Gelomics controls, where they're lacking in viscosity and a more Newtonian-like behavior compared to when you add the addition of laponite, which is these dark colors ranging from 1 to 4%. We not only see an increase in viscosity, but an increase in shear thinning characteristics, which can be further extrapolated looking at the power law regression model, looking at the N index, where something that is closer to 1 is more Newtonian-like, and something closer to 0 is more shear thinning or highly increase in shear thinning. And what we can see here is the addition of more than 1% laponite to Gelomics results in enhanced shear thinning capability. We then looked at the thixotropic test, which is applying different shear rates to mimic the extrusion of it material being extruded, and to look at its shape recovery when these different shear rates were applied. Again, looking at the Gelomics controls for 5 to 15%, we can see a lack in viscosity and a change when these shear rates are applied. And in the Gelomics 15%, which is increasing over time due to an increase in shear rate and solidulation occurring. We can then see when you incorporate Gelomics and laponite that you get these nice instantaneous recoveries when you increase laponite in concentrations of specifically more than 2%. Slide technical. And then you can see when you characterize the recovery of viscosity, we can see that laponite 2% demonstrates the increased recovery behavior compared to other groups of approximately 80% across Gelomics 5 to 15%. Next, we looked at the Young's modulus and the cross-linking kinetics, and we can see that as you increase your Young's, your laponite concentration within your Gelomics polymer concentrations, it has an influence on your mechanical stiffness. Specifically within the higher concentrations of more than 2%, in the higher polymer concentrations of Gelma 10% and 15%, there was a decrease in your Young's modulus over time, which also affected its cross-linking kinetics, which can be seen by the dark colors here in the dark purple, the blue, and in the orange as well. So LPN does affect your Young's modulus and your cross-linking kinetics depending on your concentration that you're using. And then we looked at the temperature sweep and the temperature, looking at the control, where we would like to be able to print within the room temperature conditions so we don't have to control the thermal conditions. And we can see with compared to the controls which increase within this range compared to laponite, which show where you increase your laponite concentration, there is more thermal stability within this range and fluctuations, less of fluctuations of your viscosity. So we can show that laponite therefore increases its thermal stability and reduces the viscosity fluctuations within your printing window at 22 to 26 degrees, which is ideal for what we'd like to achieve in print. So in summary for the rheology, we were able to show that the addition of laponite of 2% was the optimal concentration, which can be seen as this red line here across our key values, such as your N index, which is your shear thinning characteristic, your K, which is your velocity gradient, final Young's that you're able to achieve, the rate of change, which is your cross-linking kinetics, and also the shape recovery, which is your thixotropic viscosity ramp. So we're able to show when the addition of laponite of 2% and Gelma 5 to 15, we have a highly shear thinning capability of an index of less than 0.16. The Young's modulus can be tailorable from 0 to 60 kilopascals and has a shape recovery retention of approximately 80%. So laponite 2% was then deemed the optimal concentration moving forward to add to Gelma. We then looked at printability and different pore fiber structures at different concentrations of Gelma 5 to 15 and different nozzle sizes ranging from 22 to 27 gauge nozzles, where we assess the printability index, the PR value, so the squareness of these pores, the fiber diameters and the pore sizes, and we're able to show that different velocities and pressures induce different results and there was an optimal range within the different Gelma concentrations, and that the 27 gauge nozzle was the most optimal nozzle that resulted in decreased fiber diameter, optimal printability index, and pore sizes. We're able to show also with printing Gelma 5 to 10% with laponite 2, we can create a number of different structures and geometries, ranging from different lattice structures to droplets, to higher constructs of lattices, to cylindrical constructs, or even single fiber constructs doing these bridging techniques. So we're able to show that these are capable of printing these range of geometries and sizes of up to approximately 10 millimeters, showing that it's capable of being used for the extrusion-based printing. We then wanted to also make sure that these mixtures were homogeneous and that we were using the right buffers, as laponite has an effect in different buffers. So we looked at using millicule and heapies in these different polymer concentrations from 5 to 15%, and looking at the pH and the mean gray values to ensure that these materials were indeed homogeneous, and then also stimulating different mixing methods that would occur while we're doing this printing process. And what we're able to show is that there is quite a homogeneous structures that are able to be achieved while using the incorporation of laponite, and a homogeneous mixture is occurred. We then performed some CFD, some computational fluid dynamics, on a three-dimensional model of our nozzles, the 27 gauge, just to get an understanding and a comparison between Gelma, which is our Newtonian-like material, and Gelma laponite 2% at their corrected pressures that can be printed, and to get an idea of what their shear stresses were like. And what we're able to see is when you, with the Newtonian-like behavior of Gelma 5 to 15%, there's an increase in shear stress compared to the addition of laponite, which significantly reduces your shear stress, particularly on the outer walls, which is very important for the printing of cells, as you want them to undergo limited shear stress. And again, this can be seen when plotting the maximum shear stress over the large gradient of pressures with Gelma, which is a linear increase, and whereas compared to the laponite controls, the inclusion of laponite, the maximum shear stress is significantly less. Next, we did some biocompatibility studies, starting off in hydrogel constructs using Gelma 5% with and without lapinite of 2%, with the generation of MCF7 breast cancer cell line. And what we're able to see over the case of 7 days was in both the controls and lapinite, it was capable of forming these spheroid structures, which were also metabolically active and increased in size over time. We then translated this to more bioprinting application using a higher polymer concentration of 10% for printing purposes, replicating the same experiment and showing that when you change with the printing process that you're able to generate these large spheroid formations, which were indeed metabolically active and comparable to the controls also. So this showed that the addition of LPN demonstrated high cytocompatibility and comparable to our controls and generation of these spheroid formations using the breast cancer cell line. In conclusion, what we're able to show here is using a rheological workflow, we're able to show an optimizing strategy to optimize your additive concentration using these 4 key tests: viscosity for shear thinning capability, your thixotropy for your shape retention and viscoelasticity, cross-linking kinetics, your thermal stability to determine the printing window for temperature, and that the addition of LPN 2% enhances GELMA's rheological properties, with 10% GELMA and 2% LPN being the optimal concentration with lapinite, and that we demonstrated this high cytocompatibility. So what's next? The next step is to then further increase these biological these models by using multiple cell lines to then further actually increase this tumor complexity that is required to be achieved. I'd like to thank Gelomics, ARC CTAT, CBT, BTM group, and most importantly, my supervisors, so Professor Travis Klein, Associate Professor Laura Bray, and Dr. Christoph Meinert for all the help, and the lab members. Thank you.
Thank you, Jordan. Fantastic presentation and really a very thorough characterization of your bioink, so really well done. I think your presentation and your work really demonstrates your and also Gelomics' commitment to the development of a very scalable bioprinting solution that's also very much based on strong scientific understanding and evidence as well. So really well done. Thank you so much. So just a quick reminder to our attendees, drop some more questions for Jordan as well in the question section of the interface. We have time to address these now. So we have a few questions, Jordan, that we might just go through one by one if you don't mind. So far it's not too many, so it should only be a few minutes. One of the questions is if there are any alternative additives that you've thought about exploring in your research as well, rather than lapinite.
Yeah, definitely. We started to look at gel and gum, other different nano composites as well. But the main thing, I guess, here is because it is an industry application, there are other things that we need to consider. That if it has, while it has been done before, is there any proprietary information restricting us from building this as a product? And also the ease of use as well. So it depends. We thought that lapinite is an easy use material that we can produce on a large scale. But definitely there are other alternatives, but it depends on your printing capability that is available. And for us, we'd like to be able to print at room temperatures and control that. So temperatures that have different thermoregulations may influence this.
Next question that we had, let me just quickly go through here. How do you achieve mixing the cells into your bioink?
Yeah, so we do, we use syringes and we've got like an adaptive lure lock, which is we've 3D printed from a 2016 Michael Galensky paper who does a whole study on the different mixing methods. And it's very similar to, I think, what Cell Inc. has published with their 3D BioX, which we use, which is using 2 different lure locks, having the cells in one and your mixture and your bioink in the other, and then gently mixing them back and forth. And there's also a Cell Inc. attachment lure lock that displaces into one syringe, which can be used.
Okay, so last question we have here from the audience is, when do you think this will be commercially available?
Very good question. Hopefully soon. We've got a couple of little things that we're still working on, and we want to make sure it's a thorough product. But we'd like it to be soon. But just to make sure that it is a reliable and consistent product before going to the market.
Fantastic, thanks, Jordan. So last thing, maybe just from my side as well. So you briefly mentioned in increasing the complexity for tumor-specific models. Maybe in high level, can you comment on what that involves? Does it involve addition of multiple cell types, other ECM components, things like that? So where do you see this, I guess, platform technology moving from here forward?
Yeah, definitely. So at the moment, it's more of we've developed this standardized bioink, and we then need to further incorporate, such as, like you said, ECM components into this. But more importantly, not just generalized ECM. So for our case, we'd like to look at the prostate tumor microenvironment. So I'd like to incorporate specifically prostate proteins or DCM into this mixture. So it's a specific tissue-specific model. But this again can be applied to not just prostate, but to breast cancer or other non-cancerous models as well. And then also incorporating specific cell lines which are associated with that microenvironment, which we plan to do.
John, there's one more question. Thank you for your answer, first of all. What is the Leponite grade used in this study? Is it RD, RDS, and did you see any differences between different grades?
Yeah, so we use RD, and unfortunately with us being in Australia, Australia has specific requirements of what you can import and export, and then Leponite being a nanoparticle, we're only able to get the RD version. From what the literature says, a lot of people either use the RD and the XLG, I believe it is. And from my understanding, there isn't that much of a difference between the two. I think XLG is definitely more versatile in Europe compared to Australia.
Fantastic. Thank you so much, John. Again, thanks for your time today and for preparing this really good presentation.
Thank you so much.
I congratulate you for very thorough work, and I think you've done a really good job with your rheological characterization, really diving deep into the mechanisms of how the ink works, and then of course also your modeling and even your cell work as well. So thanks for your presentation today.
Thank you, Christoph.
All right.
Well, stop sharing the screen.
No worries. Thank you. Cool. So then we proceed to our next presentation. It's an absolute honor to have you here, Riccardo. I might just start with a short introduction, a short biography as well, and before we delve into your presentation today. So again, thanks so much for being here today. So Dr. Riccardo and Associate Professor Riccardo Levato is an Associate Professor at the Regenerative Medicine Center in Utrecht, at the Department of Clinical Sciences in the Utrecht University, and the Department of Orthopedics, University Medical Center in Utrecht, the Netherlands. He specializes in biofabrication strategies and novel biomaterials for creating tissue models and regenerative grafts with applications both musculoskeletal and soft tissue engineering. Dr. Levato's interdisciplinary team explores innovative light-based systems for biomedical research, organoid biology, and translational regenerative medicine. Excuse me. He was conferred several awards, including the 2016 Wake Forest Institute for Regenerative Medicine Young Investigator Award, the 2021 Jean Leray, I hope I said that correctly, award from the European Society for Biomaterials, and the 2022 Robert Brown Award from the Tissue Engineering and Regenerative Medicine International Society. So very well-established researcher by now, and I'm really glad to have you here. Riccardo is also serving on the board of directors of the International Society for Biofabrication. He holds a PhD in biomedical engineering and has worked in various research groups across Europe in biomaterials and regenerative medicine. And of course, Riccardo, you have also worked closely here with our group in Australia as well. That's why the confusion. So without further ado, if you could please share your screen. I'll just change the presenter view to you over now. Yeah. And we get started with your presentation.
Okay. Thank you, Chris. Thank you for the kind introduction and for inviting me here. Are you able to see my presentation now?
Yes, we can see you and hear you as well.
Okay, fantastic. So thank you again, Chris, for this invitation, for this nice series of webinars, and for the chance to introduce you and discuss with you some of the work we are doing over here in Utrecht. For those that don't know me, our lab focuses mostly on the development of different biofabrication approaches, spanning all ranges of extrusion and light-based techniques for creating more complex tissue models and regenerative grafts. And I will focus specifically on some of the light-based techniques that we are working on at the moment. Of course, we have also to develop different biomaterials that are amenable for these technologies, and that not only allow us to, of course, print a high shape fidelity structure, but also to nurture cells during their maturation process. And as Chris mentioned, we target application in the musculoskeletal arena, so mostly cartilage and bone, and some key applications on soft tissues. And I will specifically touch upon one of them in the liver case for today. The first thing I always like to remark is that bioprinting is a powerful set of technologies that allow us to pattern cells and materials in 3D. But that's only the first step of many towards achieving a functional engineered tissue. In fact, cells need to mature, need to undergo a lengthy maturation process after printing, so the printed part is not ready to be used yet, differently from what happens with conventional 3D printing, where you don't print living cells in that case. So in the case of this cartoon, essentially printing can help us to position cells in the right setting, give them the right cues, so that we have a sort of head start to accelerate that maturation process. And of course, printing is relevant because our tissues don't sit in the void, so it's always good to have ways to connect them to different perfusion systems or to other tissue types, interface them with different cell types, provide nutrients, of course, so we can print potential vasculature or the porosity that we need to ensure nutrient supply to our cells. And of course, for some tissues also needed specific mechanical support, mechanical strength, mostly for load-bearing application. And also, printing is also a way to automate the fabrication process and therefore help us to get to a more controllable and eventually also upscale to physiological relevance, physiological scale, which is something I will touch upon today. Specifically, what I would like to underline is that what is a bit of the mismatch or the gap between what we make in the lab and what happens in nature. So what you see here is a video of the development of the vascular system of a zebrafish embryo, where you can appreciate how the vessels come into being, the tissue of course grows, the organism grows as the new ramifications start to form, and this is a completely self-governed process. Whereas in most of the cases when we print cells, we have them sitting in the gels. Even at the high cell density, what you see here is an extruded filament with 100 million cells per ml. These still need to connect, find each other, and start making the right interactions in order to mature. So one strategy we looked into, it's actually a bit akin to what Jordan has presented before with the last slide, when he was mentioning that you can start to increase the biological complexity of these tumor models by combining different cell types, except that instead of combining different cell types, we start from some stem cells that form organoids, and organoids are miniaturized multicellular construct that already mimic a specific organ. I will give you an example in the case of liver engineering, where from a single stem cell, these microstructures form and different cell type already arise and organize specifically in a certain pattern inside them. And another thing I wanted to also touch upon is a recent work that we did that showed the possibility to use actually printers to pattern in 3D certain growth factors or morphogens that can then guide how cells behave over time. So the example I wanted to touch upon first is in the field of liver tissue engineering, where specifically we are interested in developing in vitro models for testing some liver functions, mostly because basically the drug-induced liver failure is the main cause of course, of liver damage caused by drugs, but also the main reason why new drugs that have been developed and reach even the market, so they pass clinical trials, actually fail into their broader use into the higher population. And that's because we don't have the right models to test this drug for patient-specific behaviors, of course. And of course, animal models we know are good, but they are not predictive enough from human toxicology. And if we try to just culture conventional hepatocytes, these quickly die in vitro, so they don't let us do long-term studies, of course. So the first thing we did was together with the group of Bart Spate at University, we started working on this pluripotent stem cells taken from the liver, specifically from the cholangiocyte duct, so the intrahepatic bile ducts. And there we have a bipotent population of cells, originally found by the group of Hans Clevers in 2015, that if cultured in 3D, form these cystic structures you see here, where depending on the media that we give them, they can either become hepatocytes or cholangiocytes, so the cells of the bile duct. Our first approach was to use a conventional extrusion printing technology. We have this very simple printed structures in JALMA, embedding these preformed organoids, which have superior metabolic activity compared to single cells, and then test toxicity on them. However, we quickly realized that if our printing blocks are not single cells, but they are relatively large aggregates, which are also quite fragile, as you can appreciate, they are hollow inside, so you have a thin monolayer of cells and then a hollow cyst inside. These are quite fragile, and the shearing through the nozzle is actually sufficient to shed cells of the organoids and cause significant death. Now, of course, these cells will recover after a few days of culture, so you can still use these models to test drugs on them, for example, but you can appreciate that this is not really ideal. So we start looking at technologies that don't involve the use of shears, and that's if you look at the different available techniques, light-based technology are the ones that at the moment allow better resolution, and none of them, of course, requires the use of nozzles, because you're just using non-invasive light to polymerize an hydrogel that encapsulates cells, and then the cells are entrapped into the geometry you are interested to produce. And specifically, we develop together with partners in Lausanne a new bioprinting approach that has been now around for a few years since we first published that, which we term volumetric bioprinting, that allows us to have a pretty good resolution in the same range of DLP printing, but a much higher fabrication rate, so you can generate centimeter scale construct in a matter of a few seconds. So the way the technology works, you see it at work here, actually, in this video on the right. That's a real-time video of a print of one of the monuments of Utrecht, the town where I'm talking from. This is the tower of the Church of Utrecht, which you see appearing here in about 15 seconds in the middle of this camera view. So what is happening here is that we are sending visible light lasers onto a digital micromirror device, which then redirect these lights onto a rotating vials containing our photopolymer and cell mixture. The photopolymer in this case is JALMA. And what the DMD does, it changes the projection with every angle of rotation of this vial, so that every angle we're sending a different 2D image, and the composition of this 2D image is generated field where the polymer crosslinks only where we want, and everywhere else it remains uncrosslinked. You can see that the process at work here, actually. Let me start the video. Yeah. Basically, we load this vial with the photopolymer again. The JALMA is thermally gelated, so there is no self-sedimentation. And with every rotation, we start sending different 2D images. Now everything is actually illuminated. So what you see here in blue is actually a fluorescent path caused by the photoinitiator itself, which in this case is LAP. And over time, the object, so in this case, this bean-shaped pancreas model appears. And what is happening there? Oh, apologies, I skipped a bit. After the object is formed, then we can just wash with PBS and then retrieve our printed part. What is happening is that basically every part that we want to have cross-linked is receive light just above the gelation threshold of the polymer, whereas every off-target area still receive light, but below the gelation threshold, so the polymer doesn't form a gel in that case. And then in this way, you can essentially get this completely sharp reconstruction of the object you're trying to make. The very first bioprint we made with this system was actually this trabecular bone model, where we reconstructed an anatomical trabecula of equine bone, long bone. And inside what appears gray in this reconstruction, there are all these magenta stained cells, which are osteogenically differentiated MSCs, through which we can then also add blood vessel cells and pericytes that start forming these capillaries attracted by these cells. If we want to use this technology further, though, especially for soft tissues, we need to go to fairly high site concentrations, and I mentioned the liver before, because we want to go in the order of tens of millions of cells per ml. And the technology relies on delivering light in a very precise manner in 3D. And unfortunately, or fortunately, actually, cells are scattering elements, so they scatter light. I say fortunately because that makes possible the use of flow cytometry as a characterization method. But of course, for printing, this is potentially an hurdle. So if you try to print either single cells or liver organoids, even already inside this sort of yin-yang structure, you get a very poor approximation of what the object should really be. So the solution we came up with is much like Jordan showed, the possibility to edit the rheological properties for extrusion. In light-based printing, we look at ways to edit the optical properties of the material. And we do that by adding a compound called iodixamol, which is a biocompatible refractive index matching agent that essentially makes some of the most scattering elements in the cells, specifically the nucleus in this case, transparent to the light that we are working with, so that we can then actually restore a solution with about 15 to 20 million cells per ml already in our structures. So with that sorted, we can see how the technology actually fares when we try to print these fragile liver organoids. And we see that compared to in extrusion, as we show already before, most of the cells after printing are shed and they start dying. And the organoids are, of course, smaller in size because we need to pass them through a nozzle, so they get fragmented. And even simple pipetting is enough, micropipetting is enough to actually partially damage these cells. Whereas in volumetric printing, we don't have any of these steps. You can just pour the organoid solution into the vials. There is no shear during printing, and therefore we have less dead cells, and most importantly, larger and better formed organoids that eventually grow better even during the culture time. It's not just a matter of size though, it's also a matter of function. So these organoids can mature faster because they start a bit further ahead in their differentiation process. And we see that only in the volumetrically printed group, we actually have polarization. So the organoids have should present some markers, in this case NDR1, which is an ion pump that in the liver pumps compound from the hepatocytes body into the intrahepatic bile duct. They should express it only inside the organoid, and that's exactly what we see here, something that is instead missing in all the other technologies and also in the matrigel control, but that's understandable because matrigel is a matrix for organoid expansion and not for differentiation in reality. Of course, we also have an expression of certain liver enzymes, but indeed more important, we have this polarization. We can then use printing as a tool to modulate the architecture around which we place these organoids, so that we can actually interact interfere with how the organoids interact with their surrounding environment. So now the cells are placed inside this two printed structure, which is here in a CT reconstruction. These are two types of gyroids, one with a very convoluted pore pattern, where you can see if we inject into the pores fluorescent beads, they follow a very up and down and tortuous path. And another one more straightforward, where the beads, if they're injected, they follow essentially a straight line. So this way we can modulate the surface area, but also how much time these beads or the solutes in the media spend inside these gels. And we tested how this can affect functionality of the organoids by injecting ammonia into the system, perfusing ammonia. What the liver normally does in our body is takes ammonia and converts it to urea, which is then eliminated by the kidney, and ammonia is toxic, of course. And we see indeed how the playing with the geometry, especially the more tortuous parts, helps the organoid to eliminate ammonia faster, so therefore to detoxify the compounds at a much higher speed, which is beneficial then to make a more relevant liver model that can then be used for testing new compounds. So this approach of using organoids is quite powerful, and we are now expanding it to different fields. We are developing, of course, this bioprinted liver organoids, but also blood vessel organoids and breast tissue derived ones, as well as engineered islets from IPS to get pancreas models. And that's something that I hope to discuss with you in the near future. The other topic I wanted to discuss is, instead of besides patterning cells, how we can also use bioprinting technologies to also pattern the chemical factors that instruct cells to perform certain tasks and certain behaviors. I showed you how you can use light for 3D printing, but essentially light-based printing can be seen more broadly than, besides as a printing method, as a way to confine photochemical reaction in 3D in a specific voxel, if you may. And volumetric printing at that allows to control these phenomena across centimeter large distances. So something that other light-based techniques cannot really do very easily. So in this case, we use gelatin norbornene-based biomaterials, which can also, of course, be photocross-linked by activation through a photoinitiator, and again, LAP in this case, and in presence of thiolated compounds generate an hydrogel. We can also think of this process for first printing, but then also to add any tile-related compound, perform a secondary printed step, so that the tile-related compound will be grafted or painted a bit in the path that we want. And if you see this example here, so the green part, which is just a cylinder with a small channel in the middle, is what we first print with the printer. And the red part, this red spiral, is not physically an object, but is a trail of growth factors that is attached in this geometry. So with this exploiting this phenomenon, we can actually generate all sorts of arbitrary chemical patterns inside our hydrogel blocks with a resolution of about 60 to 50 micrometers, more or less. And with any arbitrary pattern, as you can see from these convoluted vascular-like networks, and also combined with 3D printing. So we can print complex gyroid and then pattern growth factors in there. Now, in this case, we have a tag with a fluorescent molecule, or also this spiral inside the globe of this atlas statue. So you can combine multiple molecules as well if necessary, and exploit the powerful technology of bioprinting to make arbitrary geometries with arbitrary patterns of chemical growth factors in it. In terms of functionality, we show that using a vascular endothelial growth factor as a proof of concept. So this is one of the key growth factors in blood vessel sprouting and angiogenesis. So we print some very simple channels, lay them down with the endothelial cells, and then graft VGF on only one side of the construct. And what we can see in these channels is that indeed only the grafted region, even in absence or regardless of the absence or presence of VGF in the media, we see the endothelial cells sprouting and invading the gel, attracted by the gradient of this growth factor, something that doesn't happen in the region where the growth factor is not present. And of course, this can be quantified from imaging. And indeed, we can see that this happens both in presence or absence of VGF in the media, indicating that the grafted factor is even more potent or still potent, even in presence of abundant homogeneous distribution of this growth factor all over the construct if you apply it also in the media. So this is a powerful, interesting technology. And one last thing I wanted to touch upon is that everything I showed you is actually a single material with this chemical modification. But I mentioned at the beginning that one important aspect of tissues that actually they are not just monomaterial, but they are multi-structures. And that's a work we started tackling with one of my students, Gabriel, and the team of Thomas Jungs in Würzburg University, where we create composite materials combining light-based printing and metal electrowriting. So metal electrowriting, for those that are not familiar, in a very simple way is a sort of a combination between fused deposition modeling printing and electrospinning. So you can get very thin fibers out of thermoplastic materials, for example, PCL, what you see here. And what we did here was we printed this mechanically stiff and reinforcing meshes out of PCL, placed them into the vial of the volumetric printer, and subjected to different projections to pattern arbitrary structures interwoven with this reinforcing elements. Of course, we first have to characterize how the light is being impeded by the opaque mesh, showing. And once we know this path, we can actually correct it with the software, and then eventually obtain this vessel-like structures, which, when combined together with the meshes, showing improved mechanical properties comparable to that of a pig coronary artery. The advantage of combining these two techniques is that now we can make paths that are very complex to make by simple casting. For example, you have this channel where in red we have the hydrogel, and in green we have the mesh, we call the PCL mesh, where now we have occlusions inside the path, or we embed already a functional opening and closing venous valve inside this channel. We can also perform the printing process multiple times with the different cell types, so we can make actual multi-material constructs where we have endothelial cells in the core and the stromal cells to act as different layers of the blood vessels around. Now, of course, this is all preliminary, so there is we established the technology, and now we need to move forward towards further maturation and application. But overall, what I wanted to show you is that bioprinting can be quite powerful, especially in combination with new cell types, for example, cells producing organoids, to improve the biological function of our construct. volumetric printing and in general light-based printing are quite suitable to handle these fragile cells. And indeed, they can also be used to tune the functionality of these cells by playing with the architecture, and also to graft growth factors that can be useful for different type of applications. And this opens new possibility, of course, for in vitro models, for regenerative implants, but also for the developmental biologists, for example, that are interested in bringing 3D into their research. So with that, I'd like to, of course, thank all the people that did all the work, which is a fantastic team of students and collaborators I work with. And thank you all for your attention. I'll be happy to take any questions you may have.
Fantastic. Thank you so much, Riccardo. Really amazing presentation and really impressive piece of work there. I mean, it's amazing to see that your group has grown so much, and just this year alone, I think you've published a lot of really high impact papers that are really visionary and really dictating where the field is moving into as well. So really well done. Got a couple of questions as well. If you don't mind, we might just go through them. So I'll just read them out right now. So great talk. Thanks. Thank you, Riccardo. How do you mix the cell suspension into the hydrogel precursor solution prior to loading into the printer to enable a homogeneous distribution of the organoids without breaking up the aggregates or organoids? Right.
That's a fair point, because as I mentioned, they are quite fragile. The advantage here is that we don't need to. Let me go back to the slides there. We can just load them with the serological pipette, so that we don't have the tip of the micropipette there fragmenting the organoids. So we essentially we add our organoid suspension, which is sedimented into a Falcon tube. We add on top of it our hydrogel, and using very slowly with the serological pipette, we mix them up, and then we pour them into the glass vial.
Okay, thank you. And the organoids are generated in a conventional basal membrane extract initially?
Actually, I didn't mention that. They can be done in a conventional BME, so BME is a matrigel hydrogel. But the way we do it here is in a spinner flask bioreactor. So we don't culture them. We take the single cells, we add them into media, and we pour them in a spinner flask. And there is a little bit of matrigel in the suspension, because these cells do need laminin and collagen 4 to grow. Potentially, you could replace that matrigel for the recombinant proteins, but you know that there are still some compounds that we don't know exactly what they do, but we need them for matrigel as well. So they grow into the spinner flask and they form organoids in suspension. So they're not attached to the matrigel itself, but we do need a bit of matrigel into the media.
Cool. Thank you, Riccardo. One more question. Can you also discuss your utilization of thermal gelation to avoid sedimentation discussed in this paper? Very cool.
Sure. Yeah, so that's an important aspect, of course. So if with volumetric printing and in general with light-based techniques, if there is no stirring, one risk is potential sedimentation of the cells. Now, in this case, we don't have that issue because we are working with Gelma. So we are, or Gelma Bornini in the other case of the photografting paper. So we can simply let the thermal gelation occur, and it occurs very quickly because we can just soak the vials in 4 degrees water. So the gel essentially blocks the cells in the spot. At the beginning, when we were working with larger volume, we were even trying to steer a bit by placing a mini steering bar inside the vials to keep the organoids afloat. But in the end, we realized that the gelation is so quick that we don't need to do it. So we don't have any problem of a different distribution of cells across the height of the construct.
Thanks, Riccardo. Another question, can you comment on the utilization of orthogonal chemistry to enable multi-material volumetric bioprinting?
Yeah. So that's something that so we are investigating, and of course you can think of different chemistries like thiolene and acrylates. Of course, these are not fully orthogonal between them, but there exist other orthogonal reactions which potentially you could use for having two gels that are fully not that can be printed together, but they're not chemically bound to each other necessarily. So yes, that's definitely a possibility, and we are investigating that. It's still a bit early though for the stage of in kind of development. Outside of the bio field, there has been a work by a group in Denmark. I think it was Aarhus University that they published last year in Nature.com, a paper where they do some two orthogonal reactions to print two materials. Again, not in the bio arena, but in outside of the biomedical field. Although the two chemistries were not fully orthogonal, but it's a first indication in that direction.
Fantastic. I'm interested in the volumetric bioprinting, of course. It's a very, I guess, cutting edge technique, really fantastic work you can achieve with that. I was curious if you engineered and built the printer that you're using yourself, or is that a commercial system?
Both. So we have a commercial system, actually two commercial systems in the lab from Readily 3D, a company, a spin-off from IPFL, which is the university and the group with which we started this works back in 2018, 2019. So we have two printers from this company. Actually, the work on organoids that I showed and the work that you see here in this slide, it's entirely done with one of their printers. And then we have also our own platform that we use for more flexible / for different type of works where we want to tinker with the machinery as well, which is what you saw in the videos I put at the beginning, which we're also using for. We now have a preprint out with that printer as well. But we do use both the commercial one, which has the advantage of working very nicely, and you have everything ready to be used. And we also have, and then if you want to tinker with the optics, then we also have the option to use the custom-made device.
Fantastic. In one of your earlier slides where you were showing some demonstrations and some applications of the volumetric bioprinting, I think you had an image where it appeared that some of the cell types appeared patterned, right? So I think it was MSCs, endothelial cells in a volumetric bioprinted
this
construct.
slide, I guess.
I think it was earlier than that when you first introduced the volumetric bioprinting.
Oh yes.
And I'm just curious whether this is just spontaneous formation of the cells themselves, or if there's any way to pattern cells within different cell types.
Maybe it wasn't clear what because maybe I didn't mention it properly, but what's printed and patterned is the bone cells of the magenta you see here, which is basically following the struts of this bone model. Mm-hmm. The blood vessels and the pericytes are added later, so we seed them into the pores. Okay. So they are growing on the surface of the pores, and then what they are doing onto the surface, they are reassembling into this spontaneously into these capillaries, which we also see they are starting to invade into the gel, being attracted by the MSCs.
Fantastic. Last question I had, Riccardo, as well. Again, the combination of the melt electrowriting with the volumetric bioprinting, I think that's a very innovative solution to potentially reinforcing a soft hydrogel structure with a more sturdy, PCL or similar backbone. And particularly in the context of vascular tissue engineering, the mechanical properties of the construct are quite important. And I know you mentioned it was preliminary data that you're showing today. I was just wondering whether you've already started investigating the mechanical properties as well, in particular also the compliance of the material, because that's one of the main limitations of current techniques that are used in surgery.
Yes, so you're definitely right. So we did investigate quite extensively the mechanical properties. It's in the paper that I'm referencing here from Rosbacher et al., who was published a month ago, more or less. So we look at circumferential stress and burst pressure there, as well as the load kinetics as well. We haven't looked extensively at the compliance. We have done some cyclic tests as well. But in that sense, I think we see similar mechanical behavior to the coronaries, but I think we are not quite there yet. So that's something that needs to be improved, maybe with different type of designs, different thickness of or density of the fibers, for example. So that's something that is still, I think we're getting close, but we need something more.
Fantastic. There's one more question from the audience. Are there any issues with delamination of the PCL with the hydrogels over time in this bioconvergent print? What methods are employed to negate this?
Yeah, so we didn't observe any. So we cultured the systems up to 2 weeks actually. We have also done it under flow in a perfusion bioreactor. We didn't experience any delamination. When we look at the failure mechanics of these constructs, when we bring them to failure in our mechanical test, in the burst pressure, it's more of a punctual part, usually at the junction between the gel and the connecting system. When we look at the circumferential stress or the tensile assays, what we see is that the first thing that fails is the gel itself. So the gel is the weak component here, of course, of this composite. So we see a crack propagating into the gel, but we don't really see the lamination from the fibers. That being said, we haven't seen it, but we cannot exclude it. So it is in our experience, in our handling, the embedding is quite good. The adhesion of the gel to the PCL is quite good. It has been already investigated though in the context of non-cylindrical but more planar geometries with the composites between hydrogels and myofibers. Some ways are to improve endophilicity of the meshes, for example, by NaOH treatment or plasma treatment. Other ways are to use variation of PCLs of other polymers that can form covalent bonds with the gel, and that improves adhesion already.
Very good. We just had one more question pop up, if you don't mind. So, have you observed metabolite formation in your liver model? Maybe this was in the paper. But...
Yeah, it's in there. We look at the, in this case, we look at urea production. So we haven't looked at other compounds. In another paper, using the same cell types, we look at the metabolism of acetaminophen, which is basically the paracetamol, or the active compound of paracetamol, which in the liver is converted into a actually toxic compound. So we have seen that indeed the cells are able to perform this reaction. So we look at that. We look at albumin. So albumin is produced by these cells as well. And we look at a few liver enzymes, but not other metabolites.
Fantastic. I think that's all the questions here from the audience and also from myself. So thanks again, Riccardo. Really fantastic presentation and really visionary work you're doing there. Jordan, if you don't mind, maybe join back in. And yeah, so thank you again to our speakers. It was really fantastic presentations. Thank you, Jordan. Thank you, Riccardo. Thanks also for all the attendees. Just make sure to stay connected with us on LinkedIn and stay up to date, because we'll be hosting 2 more webinars this year. More details will be announced later. Please also make sure to check out our website. There will be more information available there as well. And of course, check out our products. So we've really been growing, particularly in last year, quite extensively. We now have our products embedded in over 21 countries worldwide, and we're getting really good feedback. So if you're interested in testing our products for your research applications, just reach out and we'll be happy to provide a free demo of our system as well. Also, if you have any follow-up questions after today's webinar, feel free to ask questions via email or any of our social media accounts as well. So with that, again, I would like to say thank you to everyone one more time, and we'll just wrap up today's session. Hopefully see you next time as well. Thank you again.
Thank you.
Thanks.
Bye.